goes. Welcome everyone to another Microseminar. It's my pleasure to introduce our speaker today, Carly Muletz Wolz. She is a molecular ecologist at the Smithsonian National Zoo and Conservation Biology Institute. And her research focuses on understanding the context of why some microbes cause harm while others are beneficial. And she uses a suite of tools in the field and in the lab setting to improve understanding of host pathogen microbe dynamics. Her findings help guide conservation action and address fundamental questions in microbial ecology. And today she'll be talking about skin defenses and microbial diversity in salamanders and frogs. Carly?
Thanks, Sarah. And thanks everybody for tuning in. I've been in my current position now for 5 years at the Smithsonian. And during that time, I've had the wonderful opportunity to continue my research on skin microbiomes of amphibians, while also expanding into other systems as well through collaborative projects, such as milk microbiomes of mammals, gut microbiomes of mammals, and soil microbiomes and coffee agroforestry. And while the latter ones are subjects of another talk another day, they all share a central theme of thinking about how microbiomes are impacting host ecology, evolution, and health, and how hosts then also impact their microbiomes. And when I think about these systems, I often think back to the early community ecologists, such as Alexander Humboldt, who wrote in a book in the 1800s, 1807, on the essay of the geography of plants, looking at how plants were distributed across mountains in Central and South America. And what he was trying to understand is, how can we explain the diversity of plants, similar to how we're trying to explain the diversity of microbes? And what his observations were that certain plant groups were distributed across elevational ranges across different mountaintops. So he started to see that these communities of plants were part of a much larger picture. And so whether we're looking at plant communities like Humboldt and present day plant ecologists, or host-associated microbial communities such as the salamander and frog system that I'll talk about today, we still want to know the same thing. What controls diversity? Now, one thing that is different in this system with the host-associated microbial communities is it's just more challenging to observe them. We can't stare at them like Humboldt did as he went and looked at these mountainsides. We have to culture them. We have to look at them through microscopes or extract their DNA. But together, we're still trying to understand these similar questions. So why should we care about them? One of the ideas is, from a basic standpoint, is just understanding how ecological communities assemble, right? Whether it's a plant community or a microbial community, knowing how these communities assemble are important to understand why the world is the way that it is, what we see on the landscape. But also we can think about it in an applied context, that if we can predict community structure and function, we can then make use of these communities, or we can predict what these communities will look like under future climate scenarios. And something that I've been thinking about a lot in the last decade in applied context is thinking about, can probiotic bacteria, beneficial bacteria, be used to stop infectious disease? To protect animals dying from infectious disease in the wild. And this has showed up in popular media, some of these articles here asking if probiotic bacteria could save an endangered frog, or could they save bats from white nose syndrome? But I've also, since working at the Smithsonian National Zoo and Conservation Biology Institute, have started to expand into asking more other applied questions, such as, can we use fecal transplants to improve the health of endangered animals in our care? But that's all setting the stage for the story that's the main focus today, which is talking about microbial diversity in salamanders and frogs. And so our story that starts with four-toed salamanders in the Appalachian Mountains of the United States. For those not familiar with the Appalachians, it's in the eastern part of the United States, is a long mountain range and is host to more species of salamanders than anywhere else in the world. And one of these salamanders is the four-toed salamander pictured here. And females stay and brood their eggs, so they attend their eggs. And an observation made by Dr. Reed Harris, which he published in 1995, was that females that stay and brood their eggs, the embryos have higher survival. Now, this behavior is somewhat common within this family of salamanders, Plethodontidae. And so others had hypothesized, dating back to the 1960s, that this behavior is to defend against predators. But what Dr. Harris saw whenever he was out in the field is that it didn't appear that four-toed salamanders were defending against predators. They didn't seem to care if a predator came or not. And so he and others began to hypothesize that maybe it was microbial defenses that these females were giving to their young, that they were, while in close contact with them, were giving them their beneficial microbes that were protecting them from pathogens, fungal and bacterial pathogens in the soil from where they lived. Now, at the same time that they began these hypotheses, there was an amphibian pathogen making headlines across the globe. This was in the late 1990s, early 2000s, and it was causing massive declines in the Americas and Australia. So while the four-toed salamander tells the story of beneficial microbes, this is one of the bad microbes that showed up. And this is an iconic image here on the left of catch per unit effort in Panama, where this pathogen had came through, work by Dr. Karen Lips and colleagues, who I'd later join Karen's lab as a PhD student, which I'll talk to you later about. But this is her work where she's studying the communities there in Panama, communities of about 60 some species of amphibians. And then when the pathogen came in 2004, there were massive declines across the board. This is showing abundance, but more than 30 species were actually lost from the site, never to be seen again. So this villain in our story is the amphibian chytrid, which is linked to declines of 200 plus amphibian species from across the globe. This pathogen that was what went through Panama is Batrachochytrium dendrobatidis, which is BD for short, or maybe short for bad. So for the rest of the talk, I'll call this guy BD. It was first described in 1999, actually here at the Smithsonian National Zoo. We're part of a team that first discovered it. And in the last decade, we've also identified another sister taxon within the same genus, Batrachochytrium salamander vorans, BSAL for short, which has a much smaller distribution. BD is found across the globe, where BSAL is predominantly found in Asia and Europe. And in both of these cases, we believe that they came from Asia, where they had co-evolved with the amphibian species there for at least thousands of years and spread through commerce to the rest of the world. And in this, not all amphibians are equally susceptible. While they're able to infect an incredible diversity of amphibians, over 200 plus species, not all are susceptible. If they are susceptible, it infects the skin of the amphibian. And the image here is where you can see that there are zoospores that develop into zoosporangia and basically clog the epidermis of the amphibian so that it cannot do important gas exchange, and the amphibian dies, sadly, as an image there on the left. And so where I joined the story is where I started my master's thesis, where I joined the lab of Dr. Reed Harris and started working with bacteria him and colleagues had isolated from the skin of a four-toed salamander. So one of them was Janthinobacterium lividum, which as a young master student, I took and I doodled on a petri dish to say, save the amphibians. And so this is a purple pigmented bacteria, which it produces vialicine, which we think is one of the compounds which kills BD, or we know is one of the compounds that can kill BD. And I wanted to ask, can we take this bacteria isolated from four-toed salamanders, put it into the environment and have it transmit to the skin of a salamander and then protect it from BD infection? So here's this redback salamander exposed to Jliv, is what I will call it for short, instead of Janthinobacterium lividum. So Jliv for short, the beneficial bacteria. And for my master's thesis, we did in fact show that. We showed that BD inhibitory bacteria, Jliv, survived in soil. So here's this first image here where we have the 2 Jliv plus treatments. We made this bacteria antibiotic resistant so that we could culture it on petri dishes. So these are colony forming units. So we could culture it on selective media. And this is how much Jliv that we could find that we knew we put into the soil would come back out. And then the ones that didn't have Jliv, the control salamanders or terrariums, did not have Jliv. And then we were able to show that we could transmit it to the salamander skin. So here's our salamander here, and then using qPCR, we were able to detect Jliv on the skin of our 2 treatments that were exposed to Jliv and not detected on our control animals. And we were able to detect that for 40 days. And I believe it was around day 12 was when we then added BD to the story, the pathogen, to see if it influenced survival or pathogen prevalence. And what we found was by having this Jliv in the soil, it reduced BD prevalence on salamanders by 50%. So this is the percentage of salamanders infected 5 days post-exposure, and 80% were infected that did not have Jliv in their soil. But if they did have Jliv in their soil, only 40% were infected. So it was a really encouraging story that we could transmit these probiotic bacteria to salamanders and reduce BD infection. But unfortunately, I wish that would have just been a success story and we would have been out using probiotic therapies across the board. But that was not the case. In the decade that's since passed, we've done a good bit of research, including one that was just published last year that I was a collaborator on this project, where adding bacteria, including even trying to genetically modify skin bacteria to produce a lot of this vialicine, this purple pigmented compound that can kill BD, did not defend Panamanian golden frogs from disease. So one of these frog species that was lost from El Cope, Panama, for instance. And so what we have is in this decade that's passed since my master's thesis is about 50-50 on success rates of probiotic therapies, that sometimes they work, but other times they don't. And why is that? And this statement here is that our inability to successfully alter microbiomes stems from a poor understanding of the factors driving microbial assembly. And that this is not unique to amphibian systems, right? We're trying to use probiotics in human medicine and they can only be so effective. There's a wide variety of systems that this also applies to. So then we have to get back to our fundamental questions. What controls diversity? And so this is what I have then spent, 8 to 10 years working on, is trying to understand this in the salamander and frog system. And one of my favorite papers to think about what's controlling diversity in a host-associated microbial community is ecosystem on a leash model that was proposed by Foster et al. In Nature in 2017. And what they proposed is we should view microbiomes as an interplay between host control and microbial competition. And so this is their ecosystem on a leash model, which is something like a mammalian gut or salamander skin. And this is opposed to models where there's just hosts that are in control, such as the bob-tailed squid that expels its symbiont daily, or symbiont control, such as the zombie fungus that turns ants into zombies, or something like the pitcher plant that's an open ecosystem where it appears as if the plant is not interacting with their microbial communities that is in the water within their pitchers. Now, I hope the authors don't mind, Foster et al. Don't mind me suggesting a couple tweaks to their models. What I think is something that I'll argue for and show you data for is that the ecosystem on a leash model should also incorporate host environment, right? The host and the microbes are all interacting within the host environment where that host is living. So factors influencing the ecosystem on a leash would be the abiotic environment of the host, and following their format host control. And they said that microbial competition was something that we should focus on. And I think that it maybe is a better idea to call it, or I would suggest to use microbial interactions, because they can be both positive and negative and not always competition. So here's our updated modified version of the ecosystem on a leash model with the ecosystem on a leash in a host environment. And we can think about this also with pictures, looking at the environment in which these salamanders are living in, and then the microbes which are living on the salamanders. And I'd like to give photo credit to my sister here that provided this image, who takes great landscape photographs. And this is going to be the outline for the rest of the talk that I'm going to use as we go through the rest of the results and story that I'm going to share with you. So looking at the abiotic environment, host control, and microbial interactions. So let's start with the abiotic environment. One of the first studies I did during my PhD was looking at elevational gradient to examine abiotic effects on the microbial community. So I went out to Shenandoah National Park and sampled redback salamanders across the elevational gradient there to look at their microbiome. And just to say, when I say the word microbiome, I am looking at the microbiome by taking a skin swab from those salamanders, extracting the DNA, and then using 16S rRNA gene amplicon sequencing to look at these microbial communities. And so unless noted otherwise, when I say microbiome, it's the 16S metabarcoding community that I'm looking at. So what we found when we looked at this elevational gradient is that skin bacterial richness and composition changed along the elevational gradient. So this first figure here, looking at richness of the microbial community of bacteria, bacterial taxa on the y-axis, and then along the x-axis elevation. So as we increased in elevation, we saw a steady increase in the number of bacteria that were present on the skin of a salamander. And then this is looking at community composition, looking at the principal coordinate axis of composition against elevation, in which we see a directional change in the microbiome across elevation. So what this looked like is, or what we found is, that environmental characteristics that co-vary with elevation influence bacterial composition. So we then did some distance-based linear models to try to figure out, well, what was it about the environment that might have been influencing the microbial community? And from a whole suite of different environmental factors we found that soil pH and substrate temperature were linked to this directional change in composition. So then what I did following that was to validate. I looked at specifically at temperature for a number of reasons instead of soil pH, but we focused on temperature going forward. And I then did an experiment bringing redback salamanders into the lab to look at how temperature and BD infection was shifting their microbial communities. And so we had a average spring temperature, an average summer temperature, and what would be an elevated temperature of 21 degrees Celsius for a salamander. And what I'm going to share with you now is just that data looking at temperature. We're going to leave BD infection out of the story until later in the talk. And with the experiment, we were able to support the findings from the field that increased temperature, decreased bacterial richness, and altered composition. So similar to that salamander that was out at the top of the mountain that had the highest species richness, the salamanders that were at the coolest temperatures had the highest species richness. And then as you increased in temperature, we lost the number of bacteria that were living on the salamander skin. And looking at the community composition here, so each salamander a dot here representing their skin microbiome, we found that those higher temperatures had the most altered microbial communities. So temperature was important. To wrap up this piece about the abiotic environment, we found that environment matters. So I hope that everybody is supportive of me putting the ecosystem on a leash in a host environment as a good idea to put into this model about how host-associated microbial communities are interacting. And for us, temperature was a critical determinant of skin microbial diversity. Okay, so then we're going to move to host control. And in the last 10 years, I've had the opportunity to go out and lead 3 field seasons in Maryland and Virginia in central Appalachian Mountains, sampling 11 salamander species from 6 genera. And each color represents a different field season and where we sampled. So black dots were a field season together in 2013. Green dots were 2018, and the blue dots were from some field surveys that we did in 2020. And I'll be sharing with you all of the data as we go through these field seasons and what we've been learning. But to make it not all about microbes, there are these beautiful salamanders that are out there that we're sampling. And these are the 11 species from 6 genera that we were able to sample over all of those field seasons. So we have the plethodon salamanders here, which are fully terrestrial salamanders. We have Desmognathus genus, which represents fully terrestrial to fully aquatic species, or predominantly aquatic species, riparian species, to Eurycia and Gyronophilus that are riparian-based species, and Notothalamus, which is the eastern newt that is aquatic but can also have a terrestrial stage, and then Ambystoma, which we sampled a larval species for that genus. And to add in our frog story here, we also started sampling a frog model species. And as much as I would love to continue always just working on wild salamander species, I'm realizing the value of model species and what they can do to help us understand this question about how microbial communities assemble. And so I've started a collaboration looking at this frog species, this frog model species, Xenopus laevis. Okay, so to start with our the first field survey that I did, what we found were salamanders within the same genus share similar communities, but the environment affects composition. So this is looking at composition, with each salamander being a dot here, and then their microbial community represented by that dot. And what we see is if you are, let's say, Shenandoah National Park at site 1 and 2, you are all in that same space together, so your communities are similar, and the shapes are representing the species. So if you're a redback salamander or a slimy salamander, Plethodon cinereus or Plethodon cylindraceus, you have a similar microbial community. But if you're sampled in a different site, you may have a different community. So within the same genus, similar communities, but still an effect of environment. Okay, then our next field surveys that we did in 2018, and then started working with the African clawed frog with Xenopus laevis. We found that amphibians from different genera have distinct microbiomes, but there's still environmental effects. So each one of these species were different from one another. But for instance, if we look at the newts that were sampled in Front Royal versus Mountain Lake, there was some shifting of their microbial community. And then the most grand field surveys that we've done in 2020 that's really expanded to a lot of species. We also have environmental samples that we took, which I unfortunately won't be able to present today. They're in the process of being analyzed. Stay tuned. But what I wanted to show you here was that salamanders, we were able to reproduce that with salamanders within the same genus, generally have similar microbiomes, while salamanders from different genera generally have dissimilar microbiomes. So I will say that it was really exciting for me because, when we find something that's different, so as a PhD student, I found that there wasn't a species effect, where we often see that in microbiome literature. And I was worried that I did something wrong, right? But here I'm able to show that, yes, indeed, the Plethodon, which are in in green and purple, still all grouped together. So there was, in fact, within the same genus, these similar microbiomes. And then there's some differences between Plethodon versus Desmognathus, which are in these orange and red colors. But it's not always the case that different genera, right? Because we see that Eurycia, the two-line salamander that's in brown, kind of overlaps with the Desmognathus. And then all the way over here are the newts. So these are these are newts that are found in ponds, and they're also grouping with the Ambystoma larvae that we sampled in the ponds. So there seems to be this riparian terrestrial environment versus ponds. And if you will notice, this was really exciting. I was just looking at this data for you guys a few days ago, that there's like a couple blue dots over here that are the newts. And I was worried we had done something wrong with our metadata, something was wrong, the species, like samples were jumbled, I don't know. But what I realized was these were actually individuals that were terrestrial adults that we had sampled at George Washington and Jefferson National Forest. So these are our pond newts that look like this. They live in the pond, they are aquatic adults. But in some cases, aquatic adults will leave the ponds and go become terrestrial adults. And then they can go back to the ponds. It's fascinating life history with the newts, but that's for another time. So there are these species effects, but yet within the same species, you could totally shift over into a different microbiome space if you change your skin texture or change where you're living. So I still have to think about this more, but it was really exciting, and I wanted to share it with you guys. And what has got me then thinking, kind of moving to the next thought about, well, what is controlling the microbiome? What are these host control factors? And this is similar in salamanders, amphibians, humans, mammals, along epithelial layers where these microbes are coming, whether it's in your gut or on your skin. Hosts produce host defense peptides, sometimes called antimicrobial peptides. And those are the host's first barrier against unwanted microbes. And so this started me to think about, well, what is it about these host defense peptides? Are they related to these differences in communities that we're seeing? And so I'll share, and this is just the beginning for us. We've been working on answering that question, and I'll share with you some of the data asking, do host defense peptides differ among species, and do they influence microbiomes? And to look at differing between species, we have some initial data looking at redback salamanders versus the eastern newt. And using mass spectrometry and transcriptomics, we've been able to identify 10 to 20 unique candidate host defense peptides per species. So these are the masses and the retention times of different host defense peptides, which are candidates. So they're larger than 1,000 Daltons and have greater than 3+ charge, are candidate host defense peptides. And when we go also and look into their transcriptomics and try to find any sequences that match putative host defense peptides, we've been looking at those protein sequences to try to figure out how many are unique and how many are shared. And in the end, what we find is there's only about 2 to 3 candidate host defense peptides that are shared between species, and the vast majority are unique to the species. And so we're moving forward with this. Hopefully there's some mass specs being run right now to try to look at what these communities of peptides are, the peptide diversity, to try to look at more salamander species and microbiomes. But we have been able to do this in Xenopus laevis and the African clawed frog. And so using mass spectrometry, we identified 8 known host defense peptide classes. So taking skin mucus soaks and looking at what the peptides were in these skin mucus soaks, and then looking at the skin microbiomes and correlating the two. And what we find are there are relationships. There's about 31 bacteria here that we found show relationships with these host defense peptide classes. And this is where the strength of the model system comes in, because we know what a lot of these host defense peptide classes are. Unfortunately, in salamanders, almost all of them are novel, and it makes it a little bit more challenging. But in this instance, we know what these classes of peptides are. And what's really cool to me that pulls out is the bacteria generally show the same trend to all of the host defense peptides. So say, for instance, Flavobacterium here in the middle, which is the orange line going up, you can see it has all negative correlations when there is a correlation with the host defense peptides. So it looks like these peptides are antimicrobial to Flavobacterium. But for instance, if you go over to Stenotrophomonas, it has almost all purple or blue colored. So positive interactions with all of these host defense peptides. So it looks like these peptides are pro-microbial in this instance. Okay, so what controls amphibian skin microbial diversity from a host control perspective, to wrap this section up? So evolutionary history matters. It seems that closely related species share more similar microbiomes, while more distantly related do not. But there's still this important caveat of considering the environment. And what may be one of the reasons that host species matter is what are these peptides that they are producing. And so we have some preliminary evidence that there's crosstalk between the host defense peptides and the host microbiome. And I'm excited to share more with you in future years. Okay, last but not least is microbial interactions. And the first thing that I want to focus on is these bacteria that are known to kill BD in culture. So the bad guy, the pathogen. We want to know, what are these bacteria, right? They have some function that we're interested in. And to do this, I identified bacteria by culturing, doing co-culturing assays, microscopy, spectrophotometry, and then sequencing to figure out what bacteria can kill BD. So go out and swab these salamanders similar to the DNA swab, but put that swab on a petri dish, isolate unique bacteria, just take the compounds that the bacteria produce, grow them up with the pathogen BD, stick them in a dish together in a 96 well plate, and figure out whether they kill BD or not using spectrophotometry. And then you can look through a microscope at them to verify, right? So this is what normal BD looks like growing. This is if the bacteria has killed all of them, and then variations. And then we can sequence these bacterial isolates to figure out what species they are, which bacteria species they are, and understand how they're related. And then we can also pair this with the microbiome data later. So I sequenced and identified 119 BD inhibitory bacteria, which belong to 17 bacterial families. And this was from that first field survey that I did. So looking at plethodon species in Catoctin Mountain Park, Shenandoah National Park, and Mount Rogers, so showing you where all of those isolates came from, the relationship of all of those bacteria, and how inhibitory they were of the of BD. And what we are working on now is trying to figure out, well, what is the metabolites, what is the compounds that these bacteria are producing to kill BD. And some preliminary data sharing with you here is that these bacteria are producing specific metabolites that may target fungi. So for instance, one of my isolates produces Virginia-factin and leucocin, which are known to be antimicrobial in other systems. And so we've got a lot more work to understand what all of these metabolites are that these bacteria are producing. So that's one avenue that we can try to understand what are the metabolites. And the other thing that we can do with these culturable bacteria is use their sequences to make inference about the microbiome in vivo in wild animals. So we've isolated these bacteria from the skin of a salamander, we've sequenced their DNA, and I have those from salamanders. And I can also use this incredible database of people that have sequenced these anti-BD bacteria from across the globe. And so we can take those sequences and match those sequences to the sequences in the microbiome data and try to make inference about what's actually happening on the salamander skin. And this is showing you data from our second field season, which has recently been published in Applied and Environmental Microbiology. Check it out. Is looking at networks of these microbiomes. So these are our 4 salamander species we sampled in that field surveys. And these are their microbiome networks. And let's look at the first one, the Redback salamanders network. So these networks are built off of, I can't remember the sample size of the redbacks, but let's say 20 redback salamanders. And what we do is look in the microbiome data to look for how many bacteria are always positively related to other bacteria across all the salamanders. And so we can then find how many positive relationships there are. So each one of these little nodes is a bacteria. And so how many times is this bacteria in a positive, like the relative abundance of this one bacteria increases, the relative abundance of the other bacteria increases across all of our salamanders. So these are all of the positive interactions. And then we can see if it's negative, the relative abundance decreases in this one and it decreases in that one. And so there's these negative correlations. And we can find out how many times we find that same bacteria with another bacteria across all of our salamanders. And this makes our microbiome networks. Now, these networks are color-coded by bacteria that are BD inhibitory that we found from cultures. So these are yellow if they are hub taxa, so taxa that are very tightly related to lots of bacteria in the network, or non-hubs, such as a dot here, that they're related to some, but they're not central to the networks. Okay, so that's what we're looking at here, and then we can look at these across salamander species. And what we see is in redbacks, for instance, there's all of these positive associations between these BD inhibitory bacteria, and they're central to the networks. And so it looks like they're stabilizing the microbiome networks here. And to the degree is similar in two-line salamanders, Eurycia, somewhat similar to Desmognathus, but quite different in newts. Newts have this kind of peculiar group up here where all of the BD inhibitory bacteria are interacting in a space that's not within the main network. And we'll get back to the story of how it relates to BD. Sorry, I'm going to go back. And so there's these positive associations that are happening, and we think that they're positive. And so what we were then able to do was to take some of these bacteria that we have cultures of and grow them together and ask the question, if you have these positive associations on the salamander skin, do you kill each other if we grew you together? And what we find is that there's evidence supports that these BD inhibitory bacteria do have positive associations, that they don't inhibit one another in co-cultures, even though they're really good at killing BD. So kind of validating what we're seeing in the networks. Okay, so now let's think about this in a microbiome. Sorry, I thought the connection broke down, but you guys are still there. I thought I missed a call from Sarah, but we're good. Okay, I'm back. Does the microbiome protect salamanders from BD at endemic levels? So let's think about this, of what's going on with these beneficial networks, these beneficial bacteria in the microbiome, and how are they relating to BD? So here's a map of the United States and showing the distribution of BD across the United States. And so pie charts showing you out of how many sampled amphibians, how many are infected with BD. And what you see that for the most part in the Eastern United States, it's low prevalence of BD. So BD is generally thought to be in an endemic level. Where in other parts of, say, in Colorado and places in California, there is some indication that BD is more at an epidemic level there. But here, where we're sampling the salamanders, it's endemic. And those 4 species that we sampled, we're asking, does the microbiome protect salamanders from BD at endemic levels? So these are our 4 species, and here is the prevalence of those salamanders. So no redback salamanders were found infected, about 20% of two-line salamanders, 40% of desmogs, and about 60-some% of the newts. So increasing prevalence of BD. So does the microbiome have anything to do with that? And I showed you earlier, there is some indication that newts have a very different network structure. But if we actually look at the BD inhibitory bacteria and their richness and abundance, we find that they are linked to lower infection probability in these animals. So here's the likelihood of you being infected with BD, whether you're a newt, or a two-line salamander, a seal salamander, or a redback. So the likelihood of being infected. And then this is the richness of, this is the number of BD inhibitory bacteria you have on your skin. So the more BD inhibitory bacteria you have on your skin, the less likely you are to be infected. And that is also similar to that if you total up the relative abundance of all of those BD inhibitory bacteria, the more of those that you have, the less likely you are to be infected. So not only do you have a high number of bacteria, but you also have a lot of those bacteria on your skin, and you're less likely to be infected with BD. So what we found here is the microbiome can likely suppress BD infection, giving high BD inhibitory bacterial diversity and relative abundance. So it seems like that the microbiome is playing a role. Okay, you remember that experiment that I showed you the data looking at across temperatures? Well, we're going to come back to that and actually add BD into the story now and complete the story on this experiment. And with this experiment, I was asking, does the microbiome protect salamanders from BD at epidemic levels? So we don't have an epidemic happening currently in the eastern United States. We have to bring these animals into the lab. And if you remember, redback salamanders in the field, we actually didn't find any infected with BD. But if we expose them to epidemic like BD loads, they can experience high mortality. So this is showing you the percent survival here on the y-axis, with less than 10% surviving at a low temperature, around 30% surviving, and up to 40 at the medium and high temperature. So really high mortality. So these salamanders, while not apparently susceptible in the field, are highly susceptible if you expose them to epidemic like BD loads. So did the microbiome have anything to do with it? I'm showing you two blots here. So the probability of dying. With one being that you have experienced mortality, down to 0 where you have survived. And this is the number of bacteria you have on your skin. So if you have higher numbers of bacteria on your skin, you are less likely to die in this experiment. And then the reverse happens here, which is kind of peculiar for this experiment, that actually if you had lower BD inhibitory bacteria, you were more likely to survive. So this got me thinking about, well, what is going on here? And it took me a long time to think through this. And what became apparent was that it was really critical to think about what's going on with the BD load, right? So is it the microbiome that's protecting the salamanders, or is it that the BD is changing the microbiome and you think it's the microbiome? So what I did in this model was to compare two models of BD microbiome interactions, asking, does the microbiome control BD load and mediate survival? Or is it that the BD load is controlling the microbiome and that is what in fact is mediating survival? And in this case, in an epidemic, what we found was that BD load is driving microbiome change. So it's driving a decrease in species richness and an increase in the abundance of these BD inhibitory bacteria. So there is some indication that BD load is selecting, is a pressure that's increasing these BD inhibitory bacteria, that maybe at endemic stages is leading them to be increased, that there is this selective pressure. But in an epidemic, it does not lead to changing survival outcome. Instead, BD load is what is driving whether you survive or die. But to add complexity, but also to highlight the importance of environment, we find that temperature is really important in this story also. The temperature is influencing BD load, that at lower temperatures, you have higher BD loads and then lower survival. Okay, to wrap up this section about what controls amphibian skin microbial diversity, thinking through microbial interactions. And we have this nice figure that is in our recently published paper looking at that I think summarizes nicely these microbial interactions. So here, for these more resistant species, we believe that skin microbiota are preventing pathogen colonization, that they have high microbial resistance to pathogen colonization. But then as the infection susceptibility increases and you're more likely to be infected with BD, that is leading to recruitment of these BD inhibitory microbes, and also potentially selecting for producing certain host defense peptides that are then interacting, and that this is leading to selection for skin defenses that impede disease progression. And so this is our model of what we think is happening, going from these microbial interactions to what is happening for the salamanders. Okay, to just wrap up here in the last few minutes to talk about, what we've covered in the seminar, and thanks again for listening, everybody, is that abiotic environment is important, that we should think about the ecosystem on a leash in a host environment, and that there are these important host control mechanisms that are happening, as well as microbial interactions. And to just summarize the overviews of those findings, that the abiotic host environment is important, and this includes things such as soil pH, and what I highlighted mostly in this talk is temperature, that this is influencing skin microbiome structure, that host species biology is important to what the microbiome looks like, and that this may relate to skin peptide diversity, and more to come on that. And that some bacteria are non-inhibitory against each other, that they're having these positive associations, but they're inhibitory against BD, and this is likely through their metabolite production. And finally, that skin microbiomes can likely suppress BD infection at endemic levels, that there's the selective pressure where the animal is not dying, and it's leading to a pressure to select for an important defensive microbiome. But at epidemic levels, it doesn't look like the microbiome can do it. So what do we do, right? So if there was an epidemic to happen, how do we move forward to protect amphibians? And I think it's really critical that we continue disease monitoring in the US and globally, looking for BD, and also for the other pathogen, BSAL, that I mentioned, which is an emerging concern for salamanders. It's not here in the US yet, and we're terrified if it does come, as what could happen to our diversity here, is because many species are highly susceptible, especially the eastern newt. And we're going to continue to learn the protective traits of these resistant species, so that the salamanders and frogs are helping each other. And I think it's really important, what I've been learning over the last few years is building these connections among resource managers, policy decision makers, and scientists. And one of these success stories is a Fish and Wildlife Service moratorium on salamander trade, in the hopes to stop BSAL introduction into the US. And you can read more about that, but that really highlights why it's important to have these connections across these different disciplines. And just to let everybody know out there, if you see a dead salamander or frog, please do report it. You can go in through the Forest Service website. There is a herpetofauna disease alert system that you can report, so that we can be out there if it does seem like an epidemic is happening. And just briefly, with the last few minutes, I just wanted to talk about that we recently received an NSF BBSRC grant looking at the amphibian skin microbial immune interface and its impact on infection outcome, where we are going to look at 3 species. Xenopus laevis and Bistum immaculatum was the best salamander model I could come up with, which is a topic for another discussion, and Atelopus sphaereus, the Panamanian frog that is highly susceptible to BD. And we're looking at how immune systems interacting with the microbiome. And just to share really quickly some of our preliminary findings, we've been able to show that we can put these BD inhibitory bacteria isolated from salamander skin, and that they can colonize Xenopus laevis skin. So this is some of the bacteria that we've put on their skin, and then sampled them 1 week later to find that they did stick, which is exciting. So we are able to manipulate Xenopus's skin in this regard. And then we've also been able to show that we can alter the resident immune cells that are in the skin of these animals, their mast cells and macrophages, and that this changes microbiome composition. So this is if you deplete the mast cells versus the controls, looking at the microbiome, and similar with the macrophages being depleted versus control. So pretty exciting, and I'm excited to share more with you all as we go forward. So I'll quickly just say, you know, it's been great to share with you thinking about what controls diversity and how we can then use these communities to understand just how ecological communities assemble, and if we can then use them to protect from infectious disease. And with that, I'd like to thank you all for listening. I'd like to thank all the people that have helped out in the field, wonderful collaborators and funding sources. So thank you.
Great, thank you, Carly. That's a wonderful seminar. It's exciting to think about, you know, protecting, not only learning about what, how the microbiome naturally can be protective, but potentially applying it to save the amphibians. So that's great. There is one question, but unfortunately, I think it's from much earlier in the talk, but maybe you can address it.
Okay.
They ask, maybe, you know, so I think this was in reference to the ISME paper with the genetically modified bacteria,
right.
Jliv. It says maybe the mechanism of Jliv helping against BD is not just the biolocin, because I think that was genetically modified to increase biolocin production. So can you comment on that at all?
Yes. So some of that work where we're trying to find out what these bacteria are producing, we've since seen that it looks like this Xanthinobacterium lividum that produces vialocin, it looks like they might have chitinase activity, and it actually may be the chitinase activity of these microbes as opposed to vialocin. And so by making a lot of vialocin, while it may kill chytrid in a petri dish, it doesn't turn it into being effective when you put it onto the amphibian skin. And then it may be these other compounds that they're producing may be more important.
Right. Yeah. Yeah. Difficult to try to go through all of either the host peptides too and all of the microbiome metabolites and see what might be most effective. I can see how that would be pretty difficult.
Yeah. It's definitely trying to like have a funnel to figure out what we're going to focus on. So at least I've got a career ahead of me to try to figure out some of this.
Let's see. So I don't see any other questions in the chat. Let's see, my most important question. So I maybe this is not the most important question, but one of the things I was wondering about is the skin of the microbiome. Is it an open system so that it's influenced a lot by the you know, soil environment? I'm just thinking of that one PCA plot where you had most of the organisms from the aquatic system over here, and then the few that you found on the terrestrial system. You know, it just seems like maybe that would be influenced by the environment that they were in substantially. And some of your work, you added some of the BD inhibitory bacteria into the soil, and they were found on the skin. So it must have some open component to it, but still be somewhat selective, I guess.
Yeah, it seems that there's a mix, that there's just this environmental component, and we do have some new data that in coming years will come out, but it looks like while the salamanders are sharing some of the bacteria with the environment, it's not the whole story. So they are selecting some of those bacteria, and there is some evidence from Andy Loud, and I remember published a paper showing that it looks like these BD inhibitory bacteria in the environment are actually in low abundances, and then on the salamander skin they're in high abundances. So it looks like they may be pulling out particular bacteria from the environment. So you have this environmental reservoir that you're selecting your microbes from, so the environment has to matter in that context, but that there's still some host selection happening.
Yeah, I can see how that would be the case, especially because you did see those differences, I think, between different genre, but collected in the same environment. So it's not completely open. So yeah, there's some selectivity. Well, we're out of time, but good luck with the new award and the research coming through. And thanks again for presenting today.
Yeah, thank you, Sarah, so much. Thanks for hosting.
Yeah. All right. Bye.